We have 1 more minute. I would like to welcome you all to the basic track, which would be running from pretty much now till 6 p.m. Yeah, I'm happy to see that there is so much of interest now in basic track and neuroscience and psychedelic research. And with this, I would keep it short and like to welcome our first speaker, Professor Dr. Gül Dölen. I would keep all the introductions a bit on a personal note today because we all know their CVs, we all know their publications. I would say I read her first paper, Octopuses on MDMA. And since that time, I've been hooked onto her research. It has gone in a long way, and now there are more high impact papers coming out from her lab. And with this, the topic of today's talk would be reopening critical periods with psychedelics, basic mechanisms and therapeutic opportunities. Professor Dölen.
Thank you. All right, well, it's Saturday morning, so I'm grateful that you're all awake and here, and I get you first, so you're fresh before you've heard, been blasted with 6-8 hours of talks. So thank you for being here. I'm going to tell you today about some work in the lab that was recently published. Well, actually, a couple papers that we've been working on for a few years now. My lab in general, we work on trying to understand the neural mechanisms of social behaviors. We've tried to take multiple different perspectives on this. We've taken I'm going to talk mostly about critical periods today, but if you're interested, we're also have some really interesting work coming out on parallel processing in the social brain, showing that oxytocin neurons and vasopressin neurons kind of encode social behaviors in two parallel pathways and different subtypes through different behaviors. And this has, really big implications for why, intranasal oxytocin keeps failing in clinical trials, why we need to take a different approach when we're thinking about autism. And then, of course, we've been working in octopus. Everybody's heard this MDMA octopus paper. More recently, we've just finished the genome of a new species of octopus, which we think is going to be the next model organism. And this genome is revealing all kinds of, really unexpected and cool potential new directions for aging and cellular senescence. And so we're really excited for that work coming up, and we'll be looking at critical periods and psychedelics in those as well. But for now, I'm just going to tell you about our critical period story. This story really started for me when I was a graduate student in Mark Baer's lab, where I worked on ocular dominance plasticity, sort of one of the most well-established and well-studied mechanistic models for understanding how critical periods work, why they close, and what are the molecular cellular mechanisms underlying that. And then my work as a postdoc really started to shift and look at social reward learning and understanding the neural circuitry under that. And we discovered a new type of plasticity when I was a postdoc induced by oxytocin. And then when I started my own lab, we took off in this direction and kind of brought the two fields together. And I'm going to tell you about that. So first, let me just orient you in case you're not familiar with the term critical period. I suspect everybody here has by now heard of that. It's not just like a special time that's critical during your development. It's actually a defined epoch, and it was first described by Konrad Lorenz in 1935. He won the Nobel Prize for this discovery in 1971, I think. And what he described is that in snow geese, 48 hours after hatching, they will form a lasting, lifelong attachment to anything that's moving around in their environment. And so this is typically their mom, but if their mom is not around, then it can be an airplane, it can be a wily scientist. And this attachment window basically closes within 48 hours of hatching. So after that critical window of time is closed, then the environment is not able to induce this long-lasting learned attachment. And so he called that the critical period. And since then, there have been critical periods described for many, many other behaviors and also some electrophysiological properties as well. So we know that song learning in birds also has a critical period. There's been a lot of work on touch and the critical period in the mouse barrel cortex for patterning the receptive fields for the whisker barrels. In humans, everybody who's tried to learn a language later on in life, I think this is more true in America than in Europe. I think you guys are still sort of open because you keep learning new languages well into adulthood. But in the United States, most people, know whatever language they learned as a child and then don't learn more languages as adults. And that's because there's a critical period for language learning. I'm going to tell you a little bit more about ocular dominance plasticity, which is the one that Hubel and Wiesel won the Nobel Prize for, and this is the one that we've learned the most about the mechanisms underlying how critical periods, are established and then get closed, and some insights into how we might reopen them. And then more recently, I think there's a critical period for motor learning, which you can reopen when you get a stroke. And that means that shortly after you have a stroke, so for about 3 months, you're able to relearn some of your motor function. And that window of time has also been described more recently as a critical period. It also closes. And so this means that, this has a very strong clinical implication. All right, so let me just orient you to ocular dominance plasticity because, there have been decades worth of work trying to understand the mechanisms underlying this critical period. And so this critical period is defined by doing a visual manipulation. So in mice and cats and ferrets, you close one eye for typically 3 to 7 days, and then you record the change in the response properties after you open that eye in the visual cortex. So it's an in vivo electrophysiological measurement. And early on in development, and sort of right around adolescence, there's this peak. This manipulation of the visual environment induces a massive shift in the response properties in the visual cortex. But if you go later out in adulthood, then that is no longer able to induce that shift. And people have tried to figure out what is underlying that, and there have been, literally dozens. I just listed a few here. I'm sorry if your favorite mechanism is not listed here, but there are literally dozens and dozens of mechanisms that have been implicated in closure of this critical period. And I'll just summarize in a big way. There are 3 sort of big ones that these all kind of fall into. One of them is metaplasticity, and that's the change in the ability to induce plasticity. It's not the plasticity itself, it's the ability to induce plasticity that changes. And that's thought to be mostly because of changes in receptor subunit composition or internalization of receptors as the brain matures. The other major mechanism that people have focused on is inhibitory excitatory balance, sometimes called EI balance or maturation of inhibition. And that's really relevant in the cortex, especially where there are these parvalbumin neurons that have really been implicated in that mechanism. And then the third one, and kind of the last mechanism really, is maturation of the extracellular matrix. So this is thought to be the thing that sort of, to just piss off everybody in the extracellular matrix field. This is sort of the grout between the tiles that allows the synapses to really get laid down and stabilized, okay? So those are the 3 mechanisms that people broadly have identified in closure of critical periods. And we've had the insight in neuroscience for a long time that if we could figure out a way to safely reopen critical periods, it would be a massive bonus for all therapeutic interventions in neuropsychiatric disease, because we have the intuition that the reason that we're so terrible at curing psychiatric diseases and neurological diseases is that by the time we get around to intervening, the relevant critical period has closed. And so neuroscientists have been looking for ways to reopen them, but the most reliable and robust ways of reopening critical periods to date have been things like deprivation, right? And who wants to cure their depression by going into, a dark room for a month? Nobody, right? And so they're not very clinically useful. In addition, when I was a graduate student at MIT, we had a sort of raging war with another lab at Harvard. And the war was kind of about like, is there such a thing as a master key? Could there ever be something that would be able to reopen critical periods writ large that wouldn't either cause amnesia, cause seizures, or cause the structural integrity of the brain to sort of fall apart. And that's because of the mechanisms that we had sort of understood as being underlying the closure of critical periods. So I have to say I was on the side that was skeptical that there was ever going to be a master key. And hopefully I'm going to convince you today that psychedelics might actually be that master key that we've been looking for since, basically almost 100 years, right? And that we just sort of stumbled into it, and this is why the psychedelics are having such broad therapeutic effects in so many different contexts, okay? And I'll explain to you why I think that's so. All right, so when I started my own lab, one of the first things that we did is we decided to take that social reward learning behavior, which is measured using this assay that is a modification of the conditioned place preference assay that has been used in the drug addiction literature for about 30 years to measure the ability of drug rewards to induce a learned association between an environment and that drug of abuse. And this group actually adapted that behavioral assay for social reward. And basically, the animals learn an association between the social conditioning and a novel bedding versus an isolate conditioning and a novel bedding. And then we just compare pre and post training, how much time they spend on each of those beddings. And if they spend significantly more time in the bedding that they associate with social, we take that as evidence that they've learned a positive association to the social condition. And like with other critical periods, when my postdoc, Roman Nardu, looked at this across development, and just so you know, this is a regression plot against something like 500 or 600 animals, males. We also have this same plot in females. Every single animal was only used for one experiment. So this is a massive amount of data to get this curve. What you can see is, like with other critical periods, during adolescence, kind of at the peak around puberty, the animals are able to robustly learn from their social environment. But as they mature, they enter into this closed state where they're no longer paying attention to those social cues. Okay. So as I mentioned, when I was a postdoc, we discovered that social reward learning is correlated to a novel type of synaptic plasticity. So this is ex vivo whole cell patch clamp recording in juvenile animals. And when we bath apply oxytocin, we induce depression in the excitatory postsynaptic currents. And that is modified by their social conditioning. And we have evidence to suggest that this is mediated by oxytocin's receptors on presynaptic terminals, it turns out, on serotonergic inputs to the nucleus accumbens. So when Roman decided to take a look at this, he wanted to know, is there any evidence that that type of synaptic plasticity changes across development, right? So if we're going to correlate the synaptic plasticity to the behavioral change, then we want to know whether or not it's similarly affected across development. So when he did these types of synaptic plasticity measurements in juvenile animals, so at postnatal day 40, once again, he was able to induce robust oxytocin-mediated LTD. But when he did it in adult animals, this manipulation had no effect, okay? And so that is evidence that oxytocin is regulated, metaplastic regulation of oxytocin plasticity. So the ability to induce it changes across development. Interestingly, even though we had previously shown that serotonin and oxytocin are working together to encode social reward learning in the nucleus accumbens, serotonin 1B induced plasticity, you can induce it just as robustly in juveniles as in adults. So it is not metaplastically regulated across development. And so this is interesting because it suggests that there's a decoupling of the serotonin plasticity and the oxytocin plasticity in the brain. And I can tell you what I think that means in the questions. All right, so we were interested in understanding whether or not we could reopen this critical period. And for a variety of reasons, we decided to look at MDMA, which is a type of psychedelic that is known to have these robust prosocial effects. This has been known for, many, many years in the psychonaut community, but the first publication of this effect was in 1978 by Alexander Shulgin, who, as you all know, is a chemist who, was working with many different psychedelic compounds. He and his wife, Anne Shulgin, who is a therapist and was using these in her clinical practice to treat psychiatric illnesses, with MDMA therapy. And we also knew from the psychonauts that, MDMA is robustly prosocial and induces people to want to get into these massive cuddle puddles. And so we were excited about this, and we thought, well, wouldn't this be interesting if this could reopen the critical period? And so what we did, and this is different from what you might have seen other people talking about MDMA, and what we did in the octopus study as well. So we're not looking at the acute effects of MDMA. Instead, what we're doing is we're giving the drug, and then we're waiting for 48 hours for the drug to completely clear, and also all of its metabolites to clear. And then we're measuring social reward learning in an animal that was treated with MDMA 48 hours later. And actually, we did a bunch of different time course stuff, and if you're interested in that, most of that data is in the 2019 Nature paper, okay? And so when we did this manipulation, what we saw is that in saline-treated animals, because these were adult animals, this social reward learning this manipulation did not induce social reward learning. But in animals that were pre-treated with MDMA, now there was this big social reward learning effect, even that similar to the effect in juveniles, okay? And this is just plotted on the regression plot just to give you a sense. So saline-treated, just like non-treated animals, no social reward learning, but MDMA pre-treated animals are responding similarly to the way that juvenile animals are responding. And so we took this as evidence that MDMA was able to reopen this critical period. Interestingly, and this is probably the most important control experiment of this presentation, but also I'm going to encourage everybody to do this control experiment in their own studies as well. This is the cocaine control. So when we gave cocaine, we were not able to reopen this critical period. And the reason that that's such an important control is because some people have made claims that what psychedelics are doing is that they're just psychoplastogens. Well, Christian can tell you cocaine is also a psychoactive drug that induces plasticity. So if we're going to understand what makes psychedelics different from drugs of abuse like cocaine, why psychedelics don't seem to have much of an abuse liability, whereas drugs of abuse like cocaine, heroin, alcohol, all of which induce bidirectional plasticity, we need to be able to find phenotypes that are different between cocaine and psychedelics. And to me, this is very exciting that if we're going to make the claim that the therapeutic effects of psychedelics are related to this ability to reopen critical periods, that cocaine doesn't do it, okay? So cocaine did not reopen this critical period, and this is just what I just said, that cocaine is able to induce hyperplasticity, and this is why I hate the term psychoplastogen. Back to the metaplasticity idea, we had also shown in this 2019 paper that oxytocin plasticity is restored following MDMA pretreatment. So MDMA induces a metaplastic upregulation of oxytocin LTD in the nucleus accumbens. And so this matches as well the behavioral data that I just showed you. Other interesting similarities between this MDMA-mediated critical period reopening and clinical uses of MDMA is that, to our knowledge, this assay, this critical period reopening mechanism, is the only one that recapitulates an important element of the therapeutic effects of these drugs, namely that they're context dependent, right? So people aren't going to raves and then taking MDMA and curing PTSD. They're doing it because they're being given MDMA in a specific therapeutic context, okay? And I think that if we look at the differences between the clinical trials for MDMA and psilocybin, yes, of course, the drugs are different, but it may be that the essential difference between the MAPS trials and the COMPASS trials is that the MAPS trials really focused on the therapeutic context as the primary intervention, with psychedelics as an assist, whereas in the COMPASS trials, the emphasis was on the drug and sort of testing the biochemical model of, depression, that it's just an imbalance of, serotonin, and that you can just restore that balance and boom, and the therapists are just there to make sure you don't have a bad trip. And the therapeutic effects were much more robust in the psychedelic-assisted psychotherapy model, and I think it's related to this context dependence. So in any case, our critical period reopening explanation, I think, is the only one that captures this feature of the context dependence of these therapeutic effects, because if we give MDMA in a social context, we're able to reopen the social critical period. But if we give it in an isolation context, we don't. And I have more evidence around this as well, which I'm happy to share at the end of the talk. All right, so when we finished that 2019 paper, basically we were very self-satisfied and we said, well, you know, this is because MDMA is social. It's different from the other psychedelics because it's got this very unique prosocial empathogenic property. But, you know, probably all the other psychedelics which don't have this prosocial property won't be able to reopen this social critical period because they have nothing to do with social. That's what we thought. And we thought that the reason that we were explaining this clinical result was that, you know, basically we were improving the therapeutic alliance, and that was the explanation. But then, as I'm about to show you, we had some more data that said, well, actually, many of these other psychedelics are also able to reopen. In fact, all of the other psychedelics are able to reopen this critical period. And so what that suggests is that even though we can differentiate between psychedelics based on their receptor binding or their acute subjective effects and classify them as dissociatives like ketamine and PCP, hallucinogens like LSD, DMT, psilocybin, mescaline. Some people like to call these the classical psychedelics. I hate that term. Because, you know, people like Saul Snyder and Alexander Shulgin were using the term psychedelic to include all of these drugs for 40 years before the chemists decided to narrow the definition around just this one subtype of psychedelic, okay? So I really don't like that attempt to try and circumscribe the definition around one mechanism that the chemists like, and I'll show you some data supporting my view in a minute. And unirogenics like ibogaine, okay? And so what this suggests, the data I'm about to show you is gonna suggest is that even though there are all these differences, that the thing that makes them one category, this ability to induce an altered state of consciousness that's shared by all psychedelics, might just be what it feels like to reopen critical periods, okay? So why do I think that? Part of it is because this is just summary data. If you want to see I'm sorry, this is not impressive. It's published in June 2023. And this is just summary data of a lot of different experiments. But basically what we showed is that there's a proportionality between the duration of the acute subjective effects and the duration of the open state. So we knew from the human clinical literature that ketamine, psilocybin, MDMA, LSD, and ibogaine, the acute subjective effects last for, you know, pretty wide range of time periods. So ketamine lasts typically 30 minutes to 2 hours. Psilocybin and MDMA's effects are about 3 to 6 hours. LSD is a little bit longer, 8 to 10 hours. And ibogaine is sort of the rock star of the group. It really lasts for, you know, 3 days, right? It's just most people who do ibogaine are like, whoa, you know, I'll never do another psychedelic again, but, you know, I don't want to do heroin again either, right? And we were interested in this because there seems to be this proportionality between the duration of the acute subjective effects and the durability of the therapeutic effects, right? So ketamine, you know, people who take ketamine for depression are required to go back to the clinic a week later and take it again, right? Whereas, you know, these durable effects that people are reporting for MDMA and ibogaine that are lasting for a year or more, right? Those are only on the longer lasting ones. And I think that's telling us something interesting about the mechanism. Anyway, so when we looked at the critical period open state in mice, it was extraordinary because just to compare, so here's the acute subjective effects in humans, here's the duration of the open state in mice. So in mice, ketamine reopens the critical period for 48 hours, and then by 1 week, it's closed. At 2 weeks, psilocybin and MDMA are open, but by 3 weeks, they're both closed. LSD reopens for 3 weeks, but then is closed by 4, and ibogaine is reopened for 4 weeks, and that's as long as we've ever measured. And actually, this is interesting because if we increase the dose of LSD by 50-fold, it does not extend the duration of the critical period open state. And so this argues against some of those experiments that people are proposing. Oh, well, just give DMT and then you can have, the massive high and have a short effect, and that'll be more clinically useful. Our data suggests that DMT will have, given as, inhaled or IV, is going to have a profile very similar to ketamine. And that ayahuasca, for example, would be more like LSD because it has this long-acting effect. And I can explain what I think are the mechanisms underlying that at the end. So what this proportionality is really telling us is that for all those drug companies out there who are hoping to make $1 million by engineering out the psychedelic side effects of these drugs, they might be interfering with the therapeutic efficacy of these drugs. The second thing it tells us is that as people who are designing clinical trials, we need to be paying a lot more attention to what happens after the patients come off the acute effects of the drug, because there is a therapeutic opportunity in these weeks following the cessation of the acute subjective effects to continue the learning process that is part of the that I believe is part of the therapeutic effect of these drugs. Okay, this is a busy slide, but I just want to summarize that basically we have a lot of evidence now to say that just like MDMA restores the ability to induce oxytocin LTD in the nucleus accumbens, all of the psychedelics, but not cocaine, also restore oxytocin plasticity. And interestingly, the time courses match as well. So if we give ketamine and look, pre-treat the animals with ketamine and look at oxytocin plasticity, 48 hours after we get a robust plasticity induced by oxytocin, but 2 weeks later we don't. And that also matches the time course of how long ketamine keeps the critical period open, whereas LSD keeps it open at both 2 weeks post and 48 hours post. And so this is very exciting, this correlation between the metaplastic result in acute slice work and the behavioral response. Interestingly, we find no evidence that psychedelics are inducing hyperplasticity. And so this is the effect that you might have heard in various papers where they're, bath applying drugs into a culture dish and showing, spine growth and massive, 40% changes in dendritic arborization. We find no evidence of that in adult ex vivo slices, right? And what I think that is a reflection of is that you can't measure critical periods in a culture dish because cultured neurons are baby neurons without any of the constraint mechanisms imposed on an adult brain. So none of the things that constrain critical periods developmentally exist in a culture dish. And so what I think is a lot of those culture dish results are just a technical artifact of doing psychedelic experiments in a dish. Psychedelics are not hyperplastogenic, and it's because cultured neurons are basically right here. They're typically 3 or 4 days, sometimes as much as 2 weeks in vitro, and they're still actively going on neurogenesis and migration, neuritogenesis, programmed cell death, synaptogenesis, and as the brain matures and inhibition matures and myelination matures and extracellular matrix gets laid down, it's much harder to induce plasticity. And that's why culture dishes have been known to most neuroscientists for a long time. You can spit on a culture dish and induce plasticity. It's just not a good way to measure plasticity, okay? All right, so I'm just gonna skip ahead now to mechanism. Pretty much everybody in this room, I'm sure, has heard of the serotonin 2A receptor. Everybody loves it as a potential mechanism, partially because, very early, in fact, the 2A receptor was discovered because radio-labeled LSD bound to a new serotonin receptor that wasn't the serotonin receptor that others were binding. This is work done by Saul Snyder, who actually started my department. And more recently, there's been some beautiful work from Brian Roth's group showing that LSD bound to the serotonin 2A receptor, induces these massive, long-lasting effects that are maybe mediated by beta-arrestin. And there have been other studies in humans showing that if you block this receptor, that you can block the hallucinogenic effects of LSD, even though LSD binds to almost every G protein coupled receptor, including all 13 of the other serotonin G protein receptors. So there's a lot of reason to think that serotonin might be the unifying mechanism. Nevertheless, we also know that these other psychedelics are binding to other transporters and receptors across the brain, so it was unclear. What we did is we used ketanserin, which is the drug that has been used in human studies, and what we showed is that LSD-induced reopening of the critical period does require ketanserin. So if we co-apply ketanserin and LSD, we do not reopen the critical period with LSD, but LSD by itself does. Similarly, psilocybin requires the 2A receptor, but neither MDMA nor ketamine requires the serotonin 2A receptor. I'm just going to go quickly. Beta-arrestin, similarly, is required for LSD reopening, but not for MDMA. It is also required for MDMA reopening, but not for ketamine and ibogaine. I will also just say, because I know there's a talk coming up later implicating TrkB in plastinogen effects, we found no effect of TrkB antagonists. TrkB antagonists do not block LSD-induced reopening of this critical period. I'm just going to skip ahead and say we also did transcriptional profiling, and what we identified is approximately 65 genes that are differentially expressed in the open state induced by psychedelics versus the closed state, and that 20% of these genes are members of the extracellular matrix and immediate early genes, which, if you recall, are some of these mechanisms that I suggested had been implicated previously in the closure of critical periods. And so what this suggests is that given this mechanistic overlap, it suggests the possibility that psychedelics are in fact this master key for reopening critical periods that we've been looking for. And in fact, there's a little bit of evidence to support this already, because ketamine, if you give it back to back to back, so like 6 times in a row, can reopen the critical period for ocular dominance plasticity. And so my lab is very interested in what the implications of this result are. And so we've been working on the critical period for stroke recovery. And we are basically trying to take the approach that if we give these animals that have, where the critical period for motor learning has closed, MDMA at this point, then we can restore the ability to learn a motor task after a stroke. And so what we're calling that trial is the Psychedelic Healing Adjunct Therapy Harnessing Opened Malleability, because clinicians just love their fancy acronyms. And we are raising funds right now to do a phase 1 safety trial for Fathom Stroke based on those results. And so with that, I'll just stop and acknowledge all the people who did the work. So this project was led by Roman Nardu, who's a fantastic postdoc in the lab, Ted Sawyer, Jared Meadows, and Mackenzie Wilkinson also contributed to this project. And Genevieve Stein O'Brien and Lyle Goff, really did the transcriptional profiling and mathematical modeling of those transcriptional studies. These are our collaborators for the stroke project. This is for the stuff I didn't really talk about. And we got reagents from MAPS and the NIDA drug program, as well as a couple of others. And these are the funding. So thank you very much.
Thank you so much. So time for one quick question. One quick question. Can the volunteers just bring the mic there or here? Sorry.
All right.
Yeah.
Students
Sorry.
priority. Okay, go ahead. Is there a student or postdoc who really had a burning question? Okay.
Two questions, quick ones. Do you answer the question why we open the critical period? And do you think it makes sense to synthesize substances similar to ibogaine, but that are better tolerated to actually use that long effect of the drug?
Yeah, so I think that the second question, yes, and I know that there's also companies trying to extend the effects of MDMA to recapitulate some of the long-lasting effects of ibogaine. I do think it's possible that, based on the ketamine results for ocular dominance plasticity, there might be an opportunity to do sort of back-to-back of shorts for people who can't tolerate that very long trip. And then the first question was what again? Oh, SSRIs. So SSRI is a little bit tricky because there is a little bit of evidence, although it's not super well replicated, that SSRIs can keep a critical period that's open for a little bit longer. And based on that, there was some interest in using them for clinical trials in stroke, and they failed. And I think that part of the reason for that is because, I mean, this is just an intuition, so it's based on a bunch of data that isn't published and some that is that what's happening is that essentially when the psychedelics, whether they're binding to the serotonin 2A receptor or to the NMDA receptor, are sort of overwhelming the synapse, because in the case of LSD and ketamine, they have extremely slow off rates, okay? And so these slow off rates are essentially signaling to the synapse, hey, something's wrong, excitotoxicity is coming, reset, 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 okay? And I think that's sort of like if you've got your computer and if something flickers, you maybe don't reset your computer. But if your screen freezes for more than a couple of seconds, you start to freak out that you're gonna kill your computer, so you hit the reset button. I think it's something like that. And I think that the ability of MDMA and LSD and all the psychedelics to essentially hit that reset button by overwhelming the synapse is critical. And I think that SSRIs don't do it because it's a slow sort of trickle that doesn't hit the alarm bell for reset. Similarly, I think that our data showing, which I didn't show, that microdoses of psilocybin don't reopen is basically getting at the same thing. Microdoses, I think, could be similar to SSRIs in their effect, but they're just not overwhelming the synapse to say reset, reopen, restore metaplasticity, break down the extracellular matrix, reorganize this synapse. That's my intuition, but I don't have hard data to prove that.